Modern optical microscopes allow us to see very small things, even cells. What if we want to go smaller, down to molecules, or even atoms at a tenth of a nanometer? The answer is high-resolution TEM. In HRTEM, electron waves are passed through a thin sample, under 0.1 micron thick, producing a diffraction pattern. This is recorded with a detector, such as a film or CCD sensor, producing a final image. How small can we see with HRTEM? We can see down to 100 times the de Broglie wavelength, which is the Planck constant divided by the square root of twice the product of the electron charge, mass, and voltage. For a 300 kilo electrovolt HRTEM recorded on a 1024 by 1024 CCD sensor, you can image features as small as a fifth of a nanometer. This is very promising, but the problem is that HRTEM records the diffraction pattern, so it's only recording the phase of the Fourier transform. We need the amplitude to reconstruct a complete image. The answer lies in how the HRTEM is set up. After the electron beam passes through the sample, the exit wave undergoes processing before it reaches the detector and becomes the final image. Here we can model and affect how the process it undergoes via aberration and defocus to understand how the phase changes before being recorded as amplitude. This processing is modeled by the contrast transfer function, consisting of an aperture function, an envelope function, and an aberration function. These terms are all based on the spatial frequency of the signal. First, we model the envelope function, which consists of terms for spread, chromatic aberration, drift, vibration, and a detector term. Specimen drift and vibration can be minimized in hardware. The remaining functions for spread, chromatic aberration, and detector damp the signal in Fourier space at high spatial frequencies and should be limited where possible. Next, we'll look at the aberration function. This is controlled by the electron wavelength, spatial frequency, and an aberration coefficient, which are known, as well as the defocus, which we can control. This is the key to reconstructing our image. In electron imaging, defocus is very important for achieving an optimum image. There are 4 types of defocus listed here: Gaussian, Scherzer, Gabor, and Licht. The aim of Scherzer defocus is to balance the 2 sides of the difference in the aberration function. When solving for these conditions to get the aberration function to almost 0, you get the following expression. However, using this defocus value has some limits. When you solve for the corresponding spatial frequency, all frequencies above that are filtered out. This gives you a really clear image, but it removes data. The next type of defocus, the Gabor defocus, is used in electron holography and will not be discussed here. Licht defocus is the solution to the data loss problem of Scherzer defocus. It uses the entire electron beam. The way it does this is by mathematically reversing the contrast transfer function. Doing that, you get the following expression. Given everything we've just learned, how do we reconstruct the real space image given only the phase that we record from the diffraction pattern? The solution is to reverse the contrast transfer function. So how do we do that? Well, one way to reverse the contrast transfer function is known as the focal series method. Recall the setup of HRTEM, where an electron wave passes through a sample, undergoes some processing, which is monitored as the contrast transfer function, goes through a detector and produces an image. If we vary the focus in the contrast transfer function and we record the image at 20 or more focus values, we can actually use this variation in focus and the resulting images to compute the amplitude and therefore reconstruct the real space image. Now that we can reconstruct the real space image and reduce the aberration through Licht defocus, we can truly achieve a very high point resolution. In fact, the highest point resolution recorded has been about half an angstrom, or 0.05 nanometers, just half the diameter of the hydrogen atom. Definitely atomic scale resolution. Thank you for listening to this quick overview of HRTEM.